This is podcast number 43. And in this podcast, we are entering chapter 12. This is where we are. We're talking again about homeostasis. This is more at a whole organism and genome level for homeostasis. Versus previously, we've been talking about homeostasis at a cellular level, and this one is going to focus on plants. So plants have a situation in that if they encounter an environment that is not amenable to life, it's a harsh environment, they can't move because they are stuck. So if you're a plant and there's not enough water arriving, or if they are facing a lot of heat, or if you live near the ocean and you have a lot of salt spray, so salt is assaulting your cells, or maybe you have too much water like you're in a flood, or extremes of temperature, you can't move to avoid these situations. So how do plants deal with it? Looking at the genetic level and looking at which genes are transcribed under different conditions, scientists have done experiments where they look at stress and for how many genes are activated or transcribed after short-term stress or long-term stress. So in general, a long-term stress activates more genes. So salt exposure for 3 hours, salt exposure for 1 hour activated 3 genes, transcribed 3 new genes under these conditions, whereas 12 hours you get 16. Osmotic pressure, so causing cells to gain water or lose water, like in flooding. Longer term exposure activates more genes. ABA is a plant hormone that it's a stress hormone, and exposure to excess concentrations of this stress hormone after 1 hour activates 1 gene, but after 12 hours, 6 genes. Interestingly, cold stress turns on a lot of genes in the short term, and long term, fewer genes, and they go in the opposite direction. So these are increases in the number of transcripts. These are decreases in the number of transcripts. And same with heat, in that short-term stress turns on a lot of genes, but long-term stress activates fewer genes and actually deactivates. You have a decrease in expression, not an increase in expression. If you look at just the number of genes in some advanced plants versus the number of genes in some advanced animals, there's a general trend that plants have more genes. So pufferfish versus golden delicious apple. Golden delicious apple has about twice as many particular type of frog. This is an aquarium frog, not a frog that you would see hopping around in the U.S. About half as many genes as soybeans, etc. You get the pattern here. In general, plants tend to have more complex genomes, higher numbers of genes. There are none that have been identified that have smaller number of genes in humans. Maybe they're using this increased number of genes to deal with stress because they can't walk away or fly away or whatever. Another sort of study looked at different sorts of conditions and different tissues of the plant. So instead of grinding up the whole plant and just say how many gene transcripts go up versus down, these are separating roots and leaves and separating short-term versus long-term exposure. And then also looking at different types of exposure. So exposure to cold, exposure to high salt, exposure to osmotic pressure. So this would be like in a flood. And this is what's called a Venn diagram, where they're not looking at individual genes, or they are, but on this graph, you only see the number of genes that change significantly with salt, for example. High salt concentration leads to a significant change in 375 different genes. And osmotic pressure leads to a change in 85 unique genes. But 106 genes go up or down, change, with either salt or osmotic pressure. So that's where they overlap. And 65 genes change that are the same genes as a change with salt, osmotic pressure, or cold. There are 65 genes that change regardless of what kind of stress it is. 375 that are unique to salt, 85 that are unique to osmotic pressure, 223 that are unique to cold. So looking across tissues, you have a similar number of genes responding to cold, whether or not it's in the roots or the leaves. And whether or not it's in the roots or leaves, there are 65 genes that respond regardless of what kind of insult it is. And but if roots are exposed to high salt versus leaves are exposed to high salt, you get a different number of genes that are activated. And this may be how easy it is for the salt to get at the cells if it's in a root and surrounded by soil versus if it's in a leaf. If after 27 hours, so this is short-term exposure, after 27 hours you get a more dramatic response in the leaf versus in the roots. Okay, but that's with genes that are activated regardless of the type of stress. But in terms of responding to the cold, it's pretty much the same. And if you look at 3 and 27 hours combined, those data sets combined, there's a big difference in osmotic pressure in a root versus a leaf, but respond to cold in the roots versus leaves is pretty much the same. And there are 42 genes that completely overlap in response to cold between roots and leaves. So even though the total number of genes is pretty similar, they aren't the same genes in what responds to roots versus leaves. So the idea with these sorts of experiments is to look at how an organism responds to a stress, how different tissues respond to the stress, and how they may use their library of genes to respond. If you do these same sort of experiments with animals, you get different sorts of responses, and in general, plants turn on more genes because they have more genes than animals do. So how do plants get more genes, and what do those genes look like? This is a golden delicious apple, and this genome of the golden delicious apple with its 57,000 genes has been sequenced. Looking at those genes, a lot of those genes are paralogs, meaning that there's more than one version of the gene in the genome. So within one genome, within the Golden Delicious genome, there's more than one version of a particular gene. Let's say the gene that catalyzes the first step of glycolysis, there may be 4 different versions of that. And that is shown in these dot plots. I'm not going to go over those. But the idea, a pattern of genes preserved on a chromosome is called synteny. So if you have gene number 1 next to gene number 42, next to gene number 89, that pattern of 1, 42, and 89 being together seems to be preserved across different genomes, and that's called synteny. Okay. In looking at the pattern of genes on the Golden Delicious genome, the scientists did this experiment, have the hypothesis that originally there were 9 chromosomes, okay, in the haploid genome, and that the genome went through several duplications and led to duplication of genomes and also fusing of chromosomes, probably during a crossing over event or a non-disjunction event. So chromosome 1 became chromosome 5 and 10 so this was an ancient chromosome 1 became chromosome 5 and 10 in the modern genome. Chromosome 2 became chromosome 3 and 11 in the modern genome, and so on. But some of these chromosomes fuse with other chromosomes. So chromosome 5 and 6 fuse to form chromosome 14 in the modern genome, and so on. Okay, so as we said before with paralogs, so if you had a version of a glycolysis enzyme on chromosome 4, now you have 2 chromosomes, you'll have 2 versions of that, and that means that you have one that you need if you need it to survive, and certainly if it was a glycolytic enzyme, you need that to survive. And the other one can accumulate mutations and maybe give the organism some advantage in a stressful situation. So because plants tend to have lots and lots of paralogs, lots of versions of a gene, they tend to have lots of switches that they can turn on in stressful conditions, and that's what you see in these sorts of experiments, where you put them under stressful conditions and you see lots of genes turned on. Okay. Another aspect of plant genomics, which is pretty cool, is the idea that they can form new species very quickly, even during a human's lifetime. So normally we think about a new species forming over millions of years by natural selection or genomic drift. But this was an example of a plant which a student or a scientist at the University of Washington noticed because he was a botanist, and he noticed that it was unusual in its characteristics, and then he worked out that this plant was a new species formed by hybridization of 2 separate species. So you have Porifolius and Dubius as separate species. They are diploid. They have a characteristic of a part of the plant anatomy called a ligule, okay? That could either be, you would think of them as a petal, but they are technically this thing called a ligule. They can be either short or long, okay? And these two, even though they are separate species, can breed and form fertile offspring that are tetraploid. And so they have all of these genes and all of these genes, all these chromosomes and all of these chromosomes. So not as a result of meiosis producing a haploid gamete, but instead fusing together cells, meiotic cells that happen to contain all the chromosomes, were still diploid, and then they fuse together and form an offspring that is tetraploid. This tetraploid offspring is fertile. So this happens in other situations where organisms can form hybrids, but the hybrid is sterile, and so that's the end of the line. But in this case, the hybrid is fertile, and it can go through meiosis and produce offspring and make offspring that look like it. And therefore, in one generation, you formed a new species that is tetraploid, looks like neither of the parents, and is fertile. So there was one species with 12 chromosomes, another species with 12 chromosomes, and they were able to mate to produce a 24 chromosome tetraploid version of a new species as a new genus, or not same genus, different species. They were also able to do that in the lab, but these were found in nature. So it's an example of how plants can use their genomes to adapt to their environment in unique ways that we normally don't think of as animals doing. Because they have lots of genes, they have lots of paralogs, lots of different versions of those genes that can respond to environmental stress, more so than is typical for animals. And they seem to be able to combine genomes and form new species in ways that we don't know examples of animals doing this, unless you can consider the endosymbiosis, where a eukaryotic cell swallowed up a free-living prokaryote, which became chloroplast or mitochondria. But plants are doing this on a whole genome level and forming new species. And this is forming a new species in a relatively short amount of time. This has happened over about 60 years, and normally we think about new species forming over millions of years. So how do they produce gametes that are diploid? Is just when they're going through meiosis, they don't have splitting of the chromosome down the middle during anaphase, and so all the chromosomes which have 2 chromatids per chromosome to go to one gamete, and then those 2 gametes fuse to form a tetraploid offspring, and then that offspring is a new species.